much for the introduction. It's great to be here. I know a lot of people in the room, but it's good to see everybody again, and I always love talking with the families. I'm going to apologize. It's a little bit of a technical talk, but if anybody would like to talk about this with me later, I'm happy to discuss. Okay, thank you very much. So I'll be discussing some recent work that we started in the lab just about 6 months ago, so this is very preliminary, but we're really excited about what we have so far. And Florian set me up great this morning to talk about some precision medicine and some projects that they're doing in gene editing. And we have sort of a similar approach that I'll discuss now. So skipping, of course, an introduction into ALD, but we know that ALD is caused by variants in the ABCD1 gene. And maybe what some people are not as familiar with is the family of ABCD transporters or ABC transporters. So this is one of the largest and oldest gene families in humans. There's 49 ABC gene families and 7 subfamilies. And of course, the ones that we're interested in are ABCD. So this is 4 subfamily of 4 genes. These are proximal transporters. And ABCD4 actually is not in the proxisome, it's in the ER and the lysosome. But one thing that I want to point out here is that ABCD1 and ABCD2 are very related. And so this is something that becomes interesting to us because we know that loss of ABCD1 in ALD causes a very severe disease. And so ABCD2 is actually overlapping such that they have a similar preferential substrate, or they can also take very long chain fatty acids, bring them into the peroxisome and metabolize those. And so this is just something, of course, natural in the body, and can we maximize on this, and can we really manipulate the system to benefit us? And this is an idea. It's not necessarily a new idea, but Stefan Kemp did some really nice work in the late 90s looking at the gene redundancy of ABCD2 for ABCD1 and published a really nice work in Nature Methods at the time, showing that ABCD2 can in fact compensate for loss of ABCD1. And so this is something that he showed in ALD patient fibroblasts. So if we increase ABCD2 using a drug, in this case he used 4-phenylbutyrate, you can actually increase the beta oxidation, you can increase the metabolism of those very long chain fatty acids within the peroxisome. And so he did this a couple different ways. He did this in mice and he did this in humans using 4-phenylbutyrate and showed gene expression. So ALDR, so this is the ALD related protein. So if we use this drug, we can actually increase gene expression of that gene in these patient cells. And that does improve beta oxidation. And so shortly after this was followed up, his work was followed up a few years later by many other studies doing the same thing, so using different drugs. A lot of studies were done studying the gene itself to figure out how we can increase gene expression of ABCD2. And people from Europe and in the US found different sort of methods to accomplish this. So certain drugs, and then later on some more elegant studies in the early 2000s using gene transfer studies showing that if we actually induce or create mice that overexpress ABCD2, we can sort of correct the problems that are associated with ALD. And so this is really nice work. The problem with a lot of these studies at the time is that the drugs or the methods that were used to increase ABCD2 are just not feasible as long-term solutions. So a lot of these drugs are metabolized very quickly, requiring high doses. A lot of these drugs had a lot of side effects that people didn't really want to deal with on top of ALD. And so I think for the most part, this idea was left alone for a little bit. There are certainly trials that went on more recently to try to address this problem. But for the most part, it's not been the answer that we need. And so fast forward about 20 years, we have an advanced toolkit now to sort of address this problem again. And I'm going to jump right into gene editing. So gene editing really is very similar to CRISPR, if people are familiar with CRISPR-Cas9. And this is just a way to take a system that sort of exists already in nature and modify that to sort of help treat some of these diseases. Monogenetic diseases such as ALD, where one gene is affected, is a really good target for gene editing. And in the past few years, even advances on gene editing have made a lot of progress. And this is a picture of David Liu. So he's someone who has sort of developed this idea of base editing. It started with cytosine base editors in 2016, and that was followed up by adenine base editors in 2017. And I'm going to talk about that here a little bit. So basically what an adenine base editor consists of is two parts. There's a DNA protein that's fused to a deaminase, and then this is the second part is a Cas9 nickase, so that's a protein that's bound to a guide. And so when these two are bound together, sort of as a complex, you can design a guide RNA. So this guide RNA is very specific to the DNA that you want to target. And so this guide will bring it to the specific piece of DNA that we're interested in, and then the Cas9 and the deaminase sort of do their thing, and I can talk about that a little bit. So this is really precision editing. So a lot of the gene editing strategies that have been used in the past, we're sort of relying on double-strand breaks, so cutting DNA and then relying on your natural systems to sort of repair that, and that can be sort of messy. It's not a really precise way to do that. And this Cas9 nickase, it's a nickase because it actually just nicks the DNA, so now it's a single-stranded break, which allows for a different system to come in and make the repairs, and it's a much more precise strategy compared to double-stranded breaks. And so very briefly, I just want to talk about how these work. So we have this complex in the guide RNA. This guide RNA, again, is complementary to the region of the DNA that we're trying to edit. And what happens is this guide RNA scans your genes, scans your genomes, finds that area that we're trying to target. The Cas9 will actually denature your DNA, so it separates the two strands into one strand, making sort of a window. Getting too much into the weeds here. My husband told me when I practiced my talk. But within this window, you can actually get that Cas9 in there, and the Cas9 will nick the DNA. And then what happens is it identifies an adenosine, which is why it's an adenosine base editor, and then that will actually remove just an amino group. And then when that amino group comes off, it turns it into an inosine. And then the natural repair mechanisms come through, change that inosine to a G, and that's read by polymerases, and now you've turned an A to a G. And that's a very simple one nucleotide change. And so he has developed a great team of people who, he's basically a factory, he's a machine, he's looking for diseases and he's looking for models to test a lot of his technologies in. And so he's been a really great collaborator for the past year or two with us. And so he's helping us really with this precision editing for ALD. And our approach here is a little bit different than what Dr. Eichler was talking about this morning that his team is working on. And what we're trying to do is to induce ABCD2. So we're trying to maximize on that redundancy of ABCD2 for ABCD1. And the reason that we like this approach is also, we heard this morning there's over 1,300 variants in ABCD1. So fixing that gene is nearly impossible for everybody. But if we can increase ABCD2, we can sort of ignore the problems with ABCD1 and get those very long chain fatty acids broken down. And again, because of this, it's also phenotype agnostic. So it should work for anybody who has a deficiency in metabolizing those very long chain fatty acids. The nice thing with these gene editors, it is a one-time delivery. This isn't something that you would need redosing on. But again, with that said, this becomes a very sort of delicate situation where you need to make sure that one-time delivery is perfect. So that is something that is still in the works. So one more variable about CRISPR-Cas9 and just Cases in general is that these proteins require a very specific sequence in order to bind and do their job, and that's called a PAM. And a PAM is usually any nucleotide followed by a GG. And what you only really realize when you're looking for PAM, what you only find out, if you're trying to find PAM sites that are close to your target region, is that that particular sequence doesn't exist everywhere that you really want it. And if you're trying to do precision editing, if you're trying to change one nucleotide in a very specific area, it becomes hard to find those regions of interest. And so Cas9 PAM variants have been designed, and these are just proteins now that can recognize a broadened sequence. So we can find these sites anywhere and really allow for much more precise editing. And so Dr. Newby and David Liu and some collaborators and independent teams now have developed these PAM variants that recognize these other sequences that allow for that much more precise editing. And so we have employed some of those within this project. It gets very complicated, as you can see. So a lot of these sequences here are very detailed sequences for how these proteins recognize the site. And so to sort of simplify, the way that I'm going to refer to them in this talk is just alternate 1, 2, and 3. So just know that these are are different protein complexes that are basically targeting the same area. And then the approach here to use these base editors is to really look at a sequence called an upstream open reading frame. And so an upstream open reading frame is a area of a gene that precedes the main portion of the gene. It precedes, the information portion that we're interested in. But what these areas are, they're highly regulated regions of sequence that maintain an ability to edit the downstream gene of interest. So these are regulatory areas that can affect expression of a gene or protein that you're interested in. And only about 50% of genes have an upstream open reading frame. ABCD2, fortunately or unfortunately, is one of those genes that does have this highly regulatory area. And so they do a number of different things. I'm not going to go through all the things that they do because I'm not an expert in upstream open reading frames. But one thing that they do is they affect translation of that downstream ORF. So the upstream open reading frame of ABCD2 affects the ability of ABCD2 to be expressed. And so what generally happens is if that upstream open reading frame is translated, it prevents translation of your main ORF. So if we read this area, basically ABCD2 does not get read. And so the idea that we're trying to do is we're trying to disrupt this upstream open reading frame to allow ABCD2 to be expressed. And so here's sort of a more simplified graphic of that same thing. What happens generally, this UORF gets read, and then we don't get translation. We don't get protein of our protein of interest. We don't get expression of our protein of interest. If we inactivate this UORF, hopefully we can now increase the expression of ABCD2. So our hypothesis is essentially that, and blocking or disrupting that UORF should increase ABCD2, and that should aid in the metabolism of excess very long chain fatty acids. And so I've been working with one of Dr. Newby's postdoctoral fellows, Sylvia Thomas. She has designed the complexes and the guides that we've been using for these studies. We've been using patient fibroblasts, so we have a number of patient fibroblasts at the Kennedy Krieger Institute from many, many years. We grow these cells to 2 to 3 days post-confluency. We've done individual electroporations per condition, so there's a lot of different combinations and a lot of different conditions that we've been looking at. We do these individually, introducing these base editors to the cells. We look at editing efficiency, so we can sequence these cells to see how well those edits took. And then we passage the cells, we grow them up, we expand them. We look at very long chain fatty acids, and then we measure or attempt to measure protein. And so we did a pilot study. It was a pretty small study. We did this back in April using healthy cells and using cells from a cerebral ALD patient. I'll just introduce you to the green and pink bars. So the green bar is normal levels of very long chain fatty acids. Very kindly, all my very long chain fatty acid data has been analyzed, run by Ann Moser herself. And of course, we know that patients with cerebral ALD have increased very long chain fatty acids, as we see in this red bar here. And so really in this study, we're targeting 2 different ORFs. We're trying to cover all of our bases here. So there's one section of this UORF that we're targeting sort of more distal, and then we have another region of that same ORF that we're targeting with a different complex base editors. And what we find in this is that if we edit these cells, so if we're comparing our purple to our pink here, we see that with some editing, we can decrease very long chain fatty acids in cerebral ALD patient cells. And if we're looking at the efficiency of that, for this, this is sort of our best condition in this case here. So the purple is about 50% of the pink bar. And then within those cells, we have about 80% efficiency in changing our base of interest. So that's a pretty nice decrease. Looking at another region within our UORF, we also get a decrease in very long chain fatty acids, about 60% editing efficiency here. And then our third condition, we actually had an increase in very long chain fatty acids. And of course, that's not what we're trying to do, but we have about a 70 to 80% editing efficiency within that region. And so, one of the things that we learned very early on from this is that we know that these UORF sites are very highly regulatory. And so any change that you're going to make here, depending on the efficiency of that change, is going to have some effect on the ability of ABCD2 to do its job, or has some effect on very long chain fatty acids. And for the most part, very long chain fatty acids, just mainly for ease, which sounds kind of crazy, but has been our primary readout for functional efficacy. And so with that, we were pretty excited. We thought this was, a really good start, and we wanted to sort of expand our study. So now, we went all in. We have 3 patient ALD fibroblast lines. Again, 2 different UARF sites, 8 different editors and guide complexes, and then 2 different buffer conditions. And so we have individual electroporations again, doing this on a second round. And then again, our readouts, of course, protein of ABCD2, trying to do Western blots to see if we can increase ABCD2, and then doing some imaging of these fibroblasts, looking at localization of ABCD2 within peroxisomes, and that's through a co-labeling with catalase. Again, we run very long chain fatty acids, lyso-PC, C26, and then look at the editing efficiency. And so this is more recent. This is end of August that we did this study. Again, we have a control and then an ALD comparing the white bar to the red bar. We have an increase in very long chain fatty acids for our patient cells. And then with a number of different conditions, looking at 4 different conditions within URF2, 3 different conditions within URF3, and then we have our delivery controls here, just looking at what happens if we put these sort of complexes without a guide into cells. Do we get some changes here? And for the most part, what we're really pleased to see is this is our original condition from our previous run. Here we're having, our levels are about 25% of our untreated. And then within URF3, we're also getting 2 conditions, which we see a pretty significant reduction in very long chain fatty acids. We do see a little bit of effect here with our delivery controls. These are all things that what it looks like when it gets back to us. It's kind of blinding to look at, but things that we make sure each of these columns here is a different base in a sequence, and then each row is a different patient condition or a different gene editor or complex. And so our blanks are blank, which is great. So we don't see any sort of changes within these areas. And if we look at UORF2, we see some editing. We're actually only targeting one of these columns, one A to a G. But this is precise, but it's not that precise. So we do get bystander effects. So there happens to be other A's in the vicinity. So we are actually getting some bystander sequencing or bystander editing in those areas. And then if we look at UORF3 in this area, we don't see a lot of editing, off target. But again, in our UORF3, we are getting editing of our main base, our main A to G, and then a little bit of bystander effects. And if we quantify this, so this is looking at across the bottom, this is URF2, our PAM, and then our 3 PAM alternates. The green bar in each of these is our target, so that's the base that we're actually trying to edit, and then the gray bars are sort of the bystander effects. So what's interesting is that this PAM condition, the very first one here, this was our best condition. So this is where we get our 75% decrease in very long chain fatty acids, and our target edit is only about 30%. What's interesting is this bystander effect on the previous A is actually quite high, so this may or may not be meaningful. And these other conditions have about 80% editing efficiency. And if we look at URF3, again, the green is our target edit, so we get fewer off-target effects here, but in general, our editing efficiency is a little bit lower. So just to summarize that and compare it back to our levels here, despite some of this sort of noise in the sequencing, we do get pretty significant decreases in very long chain fatty acids. And unfortunately, due to the nature of ABCD2 and the peroxisome and expression of a lot of these genes and proteins, we weren't able to do any sort of protein analysis at this point. It wasn't for lack of trying. I think we're going to get into that in a little bit with one of our later talks, but detecting ABCD2 with the resources that we have available has been a little bit difficult for now, but it's something that we're continuing to work on in the lab. And then just, also understanding that these URF sites are very powerful. They have very tight control over these areas. And so one of the sort of take-home messages that, we like about this is that we understand that URF modulation does something. And so it's just going to take a little bit of time to understand what those effects are and how we can sort of maximize on those to really accomplish our goals. And then, of course, understanding these effects will take time. Several variables. This last sequencing run that we did is a little bit messy. So sequencing is very expensive. We usually wait to run our sequencing until we've accumulated many experiments worth of things to sequence so that it all goes through in one run. And sometimes those are good and sometimes those aren't so good. So a lot of variables to work through in a study like this. And then, of course, our ABCD2 biochemistry is a little bit problematic. We've gone through about 3 or 4 different ABCD2 antibodies. So if anyone has a suggestion on how to do a Western blot on this, well, we're happy to talk about it. And then one other thing that we just have been working on also in parallel is looking at this in vivo. So many of us know that the ABCD1 knockout mouse is not the best model for ALD, but the biochemistry is there. So one thing that we're trying to do right now is to see if we can also take this approach into animals. So we have transfected mouse fibroblasts with similar parallel guides and editors to see if we can increase ABCD2 expression in these mice. We plan to deliver those with an AAV9, assess biochemistry, look at biodistribution for how this ends up in the mouse brain, and see what we can learn from animal studies on the same thing. And with that, I'd just like to thank the team at Kennedy Krieger, Greg Newby and his team at Johns Hopkins, and you all for listening, and I can take any questions. These, yeah, so I guess I should have commented on that. So these actually are all silent bystander effects, what we understand. I'm a little bit weary of this run in particular. We didn't get this sort of noise with our previous attempt on this, so I don't want to take too much away from the sequencing data, but those other A's that precede our target A should be silent changes, yes.
Okay, sorry. I think this is fantastic work. Thank you so much for doing it. I have 2 questions, if you'll allow me.
Sure.
The first is, how confident do you feel that, I know the protein expression is really hard in this particular case, how confident do you feel that protein expression is going to fall in line with that VLCFA levels, knowing that VLCFA is not always reflective of disease control?
Right. That's a good question. That's always something to keep in mind. I mean, a lot of studies have been done that just show no real relationship with very long chain fatty acid levels to anything else. I think I don't know how well they would line, but I would feel more confident in the ABCD2 levels than in the very long chain fatty acid levels being meaningful. I don't like having one readout for a study, so I'm really looking forward to getting those the protein levels back. But I think we just have to see how it kind of shapes up, and hopefully the protein will be meaningful. There's a lot of variables here with just expression of ABCD1 versus ABCD2 and cell types and location and things like that, so we just kind of have to see how it unfolds. And one thing I also want to mention that I meant to is that, in addition to the fibroblasts, we have patient iPSCs, we're developing organoids, so I think, in addition with the organoids and the mouse, I think it's going to be a really good way to sort of understand a little bit more of that relationship.
I think I'm sure we'll have some more information from you next year. And then just thinking forward, do you ultimately see this as an editing platform for AVN, or sprinkle disease, or both? How do you see this? What is sort of that treatment?
Yeah, that's a good question. In a perfect world, you'd be able to. This would be a safe thing to introduce to somebody. The mechanisms to introduce it to somebody would be safe, and you could say, oh, you have a mutation in ABCD1, let's give this to you right now, and it doesn't matter what your prognosis or your projection would be, that this could help you. I don't know if we're going to get to that. It would be nice if it could apply to AMN and cerebral ALD, certainly, and for women as well. But one thing that I really hope to glean from this study is just to get more of an understanding on the role of ABCD2, the upstream open reading frames, ways to induce or modify ABCD2, and then perhaps we can take other approaches like ASOs or other things to target that, but use this as our very clear understanding of how to get there. Hi.
Hi. Very impressive and exciting work, so congratulations. And looks like the field is moving forward. I have a question, more of I'm not trying to correlate the very long chain fatty acid with clinical response and all of that. We all understand that. But I was just curious about mechanics. In one of your experiments, there was an increase in the very long chain fatty acid in one of the constructs. So I was just curious, what happened?
I don't know. Yeah, it's a good question. It might be something that with enough iterations of this, we can figure out, if there are certain areas of the UOR for certain guides or combinations that, have that effect. Maybe the protein levels will kind of glean some, shine some light on that. I don't know. We've also, understand that depending on the nature of the cells, just the density of the cells, how long they've been in culture, this sort of thing, the very long chain fatty acids can be sort of different. So there's some variability there. I can't take anything from this, but we have an eye on it. It's not our goal to increase very long chain fatty acids.
Another question.
Yes. I'm sorry, ignore this part of the room.
No, that's okay. I'm just in this Epson. I really like that you're looking at other PAMs and targets, but I'm wondering, since this is a rather large chain family that probably has a lot of homologies due to, the evolutionary gene duplication events, have you looked with MySeq for off-target effects and other upstream regulatory regions in the other genes in the family to maybe explain why in the wild type there were these changes?
Right. That is a good question. So we haven't done that. We haven't looked at that right now. I think the plan is to, once we sort of, for example, if we move forward with some of these sites and editors that look the most promising, we would do a much more thorough analysis of that. I think, actually, if we really wanted to study some of these UORF sites and the effects, perhaps having a whole study where we, take these variables and look in depth to figure out why some go up and why some go down, could be in itself a good study. But at this point, I don't have that much information about the other genes.
So,
Hi.
Yeah, really cool stuff. I was just curious what you think about where the gene editing delivery system needs to be delivered to in the body to make the biggest impact. If you could comment on that.
So I guess something like this for ALD, it would make the most sense, I think, for an intrathecal type delivery. I think that's maybe not for the mice. For the mice, we'd probably do more of a cisterna magna or an ICV injection type thing. But I think for, I'm not a clinician, but, I imagine for ALD, where spinal cord and brain is a big part of it, we would want to do something intrathecal.
I have a follow-up to that.
Yes, please.
The approach of 89 in newborn to correct a wide CNS plus endothelium, which is relevant for cerebral ALD in this case, and who knows if you get to the adrenal gland, will be excellent. The issue that we have is the justification to do the high-risk gene therapy in newborn, where these therapies have shown there are 7,000 babies have been treated now with SMA, with 89. So, are we ready for that?
Right.
We have the biomarkers and the stuff. This is a question for the whole community. But the approach that you're taking on starting to see what are the possibilities of in vivo and the off-target real biological effects, it's going to be so critical.
Yeah. It's got a long way to go still, but yeah.
If I can ask a technical question, just on, you said you were using David Liu's Cas9 base pair editor, and as we know, the AAV cargo is 4.7 kb. His base pair editor will not fit an AAV right now. Have you been looking at some of the smaller Cas molecules or other delivery approaches? Because otherwise, this won't get there.
That's a Greg newbie question. I don't have an answer to that.
Right, you may want to get some guidance because there's smaller Cas molecules that may fit an AAV.
Right.
Hopefully we can get some better delivery over time, but if we're limited to AAV, we've got to fit in 4.7 kb.
Right.
It does, but she was using Cas9. Yeah, but we've got to use the smaller. It's just it would be good to using constructs that will fit in AAV now, so all the data we generate can be used.
Right.
All right, thank you.
Thank you very much.